In incomplete dominant, the main character was not, or the dominant character was not able to suppress the recessive character, so they mixed and formed one more character. Here phenotypic and genotypic ratios are the same. What was the ratio? It was 1:2:1. O cannot have any antigen because it will need to have the antibody A and B. Hello everyone, a warm welcome to this session of concept class for class 12th biology. I'm Dr. Divya, biology faculty. So in this session, that is of chapter 4, principles of inheritance and variation, we will learn about non-Mendelian inheritance. That is, we'll learn two concepts: incomplete dominance and co-dominance. So in the previous session, we had learnt about the laws of Mendel, that is law of independent assortment, law of segregation and law of dominance. So in this session, we learn about non-Mendelian inheritance. So talking about non-Mendelian inheritance, so there were some drawbacks that is in Mendel's theory. So Mendel had told that, like when we take a plant, say for example, which was his first experiment. According to his first experiment, he took a pea plant showing contrasting characters, wherein he took one pea plant which was taller in height and one pea plant which was dwarf, and he crossed over between them, and he said that all the resulting progenies that are there will be tall, or either tall or short plants will get expressed in the F2 generation. But according to these, that is incomplete dominance or beat co-dominance, what incomplete dominance states is that it is not that always the main characters will get expressed. That is, it is not that always the plant will be tall or it will be completely dwarf, but also we can get plants which are intermediate of height. So that is what this incomplete dominance means. So it is an inheritance in which heterozygous offsprings which are there, they will show intermediate character between two parental characters. As I said, it is not that you make a cross between a tall plant and a dwarf plant. When we make a cross, of course, in the resulting progeny, we can get tall plants, we can get dwarf plant, and not just that, we can get a plant which is of intermediate height, that is between the tall, not even dwarf, that is of shorter height. So that is what this law means. So in NCERT textbook, they have taken the example of flower color in Snapdragon. So remember the Snapdragon, it is very important question that usually comes up in the exam, that is incomplete dominance, wherein explain incomplete dominance using Snapdragon. One more name for Snapdragon is dog flower. One more name for Snapdragon is Anthurium species. So if they give this about dog flower or Anthurium species, don't get confused what it is. All are one and the same. It is the name for Snapdragon itself. Or we can take the example of Mirabilis jalapa, which is also called as 4 o'clock plant. So we'll take this example to explain incomplete dominance. In the textbook, they have taken Snapdragon in NCERT textbook. While explaining here, I've taken Mirabilis jalapa. So Mirabilis jalapa, which is also called as the 4 o'clock plant because it usually flowers at that particular time. So this plant, wherein we have taken the male plant and the female plant. So male will have, this is the male which is red in color. This is the female which is white in color. So what we will do, we'll take a male plant and we will take, if you get confused, instead of writing R1, R2, we can take it as capital R and small r. So that is easy to understand. So what am I taking here? First is parental generation or P generation. So in parental generation under male, what have we taken? We have taken red flower, right? In female, we have taken white flower. Now we'll write the genes for these. So for red it is or factors. What are the factors here? For red it is capital R. For white it is small r. So the resulting gamete that we'll get is RR. So this is the F1 generation. So in F1 generation what we will get? We will get RR. So RR means it is nothing but it is what we should think. It should be red because R is dominant. Dominant character will get expressed first. But no, what did they find out? They found out it produced pink flowers. That is an intermediate, neither red nor white. In between red and white, when we mix red and white, what will get? Pink, right? So intermediate it was found. So therefore, incomplete dominance. The dominant flower was incompletely dominating the recessive flower, wherein it could not completely dominate the recessive flower. Therefore, an intermediate colored flower got produced there, which was that pink. Now next, what we are supposed to do, because F1 generation will not give us the accurate result, so we have to self between the F1 generation. So RR into RR. Now, if we get confused, what we need to do? We need to put a Punnett square, right? So this Punnett square, so wherein we will take male and female. Now male I have to write here. So what is the male one? It is capital R, small r. The female, it is again capital R and small r. Now we'll have RR, RR, capital R, small r and RR. So what is this? This will be, these are having both dominant genes here. So it will be red. The flower will be red. Now here, intermediate. This cannot completely dominate the recessive R. So it will produce pink flowers. This also will produce pink flowers. Now this is small r. It will be small r. It is nothing but white flowers itself. Next, moving to, this is what did we get here? This is the F2 generation. After selfing between the F1 generation, we got this. Now we have to calculate the phenotypic and the genotypic ratio. So phenotypic, what all we are supposed to take? Red flower. Then we'll take pink, because second is the pink, that pink and white. So how many red? 1, right? How many pink? 2. How many white? It is 1. So what is it? 1:2:1. Now we have to take genotypic. In genotypic, we have to consider the genes. Capital RR is 1, then small r is 1, and one more that we have is small r. So ratio again here is 1:2:1 itself, because capital R, 1, 2 heterozygous we have, and again homozygous dominant we have 1, heterozygous we have, that is incomplete dominance we have 2, and we have homozygous dominant we have 1. So therefore 1:2:1. So in incomplete dominance, in the F1 generation, that is in the F2 generation, both the phenotypic and the genotypic ratio will be the same. So that is what is stated here. No, here phenotypic and genotypic ratios are the same. What was the ratio? It was 1:2:1. So that is what we got to know here. So this means that R, or this one, this R. So what is this R? It is red. Dominant, right? Was not completely dominant over the recessive R, which was white. So therefore what happened? Red and white mixed up to produce pink color flowers. So that is called as incomplete dominance. Okay. So not just this example, we can take the height of the plant, any example we can take. So in the NCERT this is taken. So that is what we have done here. Same thing is followed for Snapdragon. It's the same thing. Only thing is here I have taken Mirabilis Jalapa. In the textbook it's mentioned as Snapdragon. That's it. So this is about incomplete dominance. So next we'll move to codominance. So codominance means both the characters are getting expressed here. That is, both the characters are equally important and they are getting expressed. In incomplete dominant, the main character was not, or the dominant character was not able to suppress the recessive character. So they mixed and formed one more character. Okay. Wherein red mixed with white to form pink. But in codominance, there is no mixing of characters. Both will get expressed and both are important. For this, ABO blood group in human is one of the very best example we can take. In that too, the people having AB blood group, whose blood group is AB, they will actually have this codominance because their A is also expressed, B is also expressed. While calculating, I'll show you about that. Okay. So what is codominance? It is the inheritance in which both the alleles of a gene are expressed in a hybrid, meaning both are expressed and both are equally important there. It is not that one is dominant and the other is dominant. Both are dominating and they are existing together. Okay, that's called as codominance. So example is ABO blood grouping in human, and this ABO blood group in humans are controlled by gene. Which one? Gene I. So it will have IA, IB. So these are the genes, that is IA and IB. There is one more, small I. So that is for O. So how many genes are controlled by how many alleles? So these are the alleles which control these genes. It is controlled, the gene is controlled by these three alleles, that is IA, IB and I. And the genes are represented by I. In this, these two genes that are there, they will produce a sugar. So they are nothing but, we're talking about the blood, right? And blood is nothing but RBC, we're talking about red blood cell. It is nothing but a cell. So it is covered by plasma membrane. In that plasma membrane, sugar will be produced. In which? In IA and IB. But in I, it has no sugar. No sugar is produced. So therefore, these two will be dominating this. So when they have, that is in this allele, say for example, IAI is expressed. That is in the progeny, if IAI is expressed, no, then IA will be the dominant one. The blood group will be A. If IBI is expressed, no, then the blood group will be B itself, because this is recessive one, okay. But if II is expressed, then it will be O blood group. And if both IA and IB, it's like this. So it's easy to understand. Say for example, there are three alleles, no, and often they are in pairs. So if they have, say for example, if the mother is IA and IB is there, that is mother is IA and father is blood group B, then it can be AB will get expressed. The blood group will be AB. Or say for example, now this is called as codominance. This is one of the best example because both A and B is equally getting expressed here. Or say for example, if IAI is expressed, then the blood group will be A. If IBI is expressed, the blood group will be B. And II is expressed, the blood group will be O. This is how it is, okay. So we'll learn about this. Before that, we'll put a table of antigen, antibody and the blood groups and all that for you to recall how, so that you'll understand better. So we'll put a table here. So in one end, I'll write the group, that is the blood group. Then we'll write the antigen for that. And we'll write the antibody. And we'll write the genotype for this. Genotype. Now, if the blood group is A, the antigen will be A, but the antibody should be the opposite of A. It will be B here. First, we'll write this and then I'll tell you why it is so. So, it will be B here. Then, next is blood group B. So, here it will be B, it will be A. Next is blood group AB. It will be A and B. But here nothing will be there. Next, blood group O. Here nothing. Here it will have A and B. So why is this? Because if the blood group is A, antigen is A, and if antibody is also A, then clotting of the blood will occur. So therefore it will be opposite of the antigen, whatever antigen it is having, it will be B. Now here blood group is B and therefore antigen is B. It will have antibody A. If B occurs here, then clotting of blood will occur. So therefore, antibody will be A here. So that is why transfusion of blood is always specific. Next, A and B, it can't have any antibody. Why? Because here, if A and B is there, both A and B is the antigen which is present in the blood group AB. Now, if we take any antibody, I can't take O also as the antibody here. Not possible. That is, we can't consider that like that. But A and B, it is the antigen. So therefore, it neither can have A antibody or B antibody, because any one of the antibodies, it has also coagulation or clotting of blood will occur. Now, O cannot have any antigen because it will need to have the antibody A and B. So A and B, if it is having here, then here it can't have the antigen. So if it has A and B antigen here, then clotting of blood can occur. So very well our bloods are designed, right? So that is how the grouping will happen. Next is talking about the genotype. What are the genes responsible for these particular blood group? So for A blood group, the gene is IA. Genotype, it is genotypes, it is always represented in pair. So I have to write it 2 times. That is, this is homozygous, and one more it will be IAI. So therefore, what is getting expressed here? Only A is getting expressed. So whichever individual is having this A blood group, which is the gene responsible for them to have A blood group, it is IA, because these are the genes. A is the allele. It will carry the allele IA, and small I, because here small I will not get expressed. Now talking about this, it will be IB, and I. Next here it will be both IA and IB. It can't have small I here, because IA and small I if we write, only A will get expressed. IB and small I if we write, only B will get expressed, right? This is the best example for codominance. Now this is codominance, wherein both are getting expressed. IA and IB both are equally getting expressed. Now in here it is only the small I. It can't be capital IA and I, small I. Again, because if it is IA and I, it will become blood group A. So therefore, they will not have any, they will have only this recessive gene, that is small I. This is how the genotype is written. Now we have understood this. So let's go further talking about codominance. So what did I tell you? The plasma membrane of the RBC will have sugar molecules, and these sugar molecules will protrude from the surface of the plasma membrane, and that production of the sugar molecule is controlled by the gene. So therefore, genes capital I will have sugar molecule, small I will not have sugar molecule. So the gene I has, which is the gene 3I. I has how many alleles? So this is the gene I. I'll write it like this. How many alleles it has? It has A, that is IA, then it has IB and I. In this IA and IB, these two are the ones which will produce the sugar. I is the one which will not produce the sugar. So this doesn't, IA, that's why I've striked it, meaning it doesn't produce sugar. So this is about the gene I. And IA and IB produces a slightly different form of the sugar, whereas allele I that is there does not produce. So IA and IB allele produces sugar. Small I will not produce any sugar. Where when IA and IB are present together, they both express their own types of sugar. This is due to codominance. So that is why IA and IB both gets expressed equally in the individuals having AB blood group. So we have taken here what are the genes. So say for example, this will consider the male and this will consider the female. So we've taken the genus IA and IA. So here what will be the blood group? It will be A blood group, right? You can see the shadings here, A blood group. IA and IB, it will be AB blood group. IA and small I, A blood group, because I will not get expressed here. So therefore, here it will be again AB blood group. This is B blood group. This one, it will be B blood group. This one A, this one B, and this one will be O. So therefore, if a progeny has to have O blood group, then the mother and the father should also have O blood group. So if the mother and the father has a blood group, the child will also have A blood group. or if they have A and O blood group also, say for example, if the father is a A blood group and the mother is the O blood group, then the progeny will have the A blood group. It is like that. So that is how blood grouping is done to find out what blood group the progeny will have. So this is about, now here this AB that is there, this is codominance because both A and B are getting expressed forming AB blood group, right? So this is about codominance. So I hope you understood this session well. So in the coming session, we'll talk about chromosomal theory of inheritance. So far, we were just talking about factors, alleles, genes and all that. Where are all these present? They're present in the chromosome. How did they get to know after the advent of the, but that was during Mendel's time, microscope was not there. But later on, after the advent of microscope, that is when they got to know that genes are inherited through the chromosomes, because that is the whatever gametes are there in the, that is chromosomes, and whatever in the gametes chromosomes are there, that will go to the progeny, and those chromosomes are the ones that will carry the gene. So that again will depend on how far the gene is placed in the chromosome and how close the genes are placed in the chromosome. That is called as linked and non-linked genes. All that we'll study in the next session while talking about chromosomal theory of inheritance. In that, we will also learn about linkage and recombination. So, all this Mendel could not explain when linkage and recombination, that is how close the genes are, how far the genes are, and once when crossing over takes place, the genes will recombine and they'll shift their position to the chromosomes. All that will lead to an effect in the progeny. That is, the progeny's character will change. All that Mendel could not understand because microscopes were not there during this time. But still, because of his contribution to explaining how the characters move from the parents to the progeny, he is called as the father of genetics. So all this we'll study in the next session. So see you in the next session. Thank you.